Welcome to the Wild Side. This is your host, Kight, and that was Bob Marley with Three Little Birds. And on this week's episode, I am going to talk about a recently released scientific paper entitled Mercury Reduces Avian Reproductive Success and Imposes Selection, an experimental study with adult or lifetime exposure in zebra finches. And I know that's quite a mouthful, but it is actually quite an interesting paper, and I'm not just saying that because it is partly authored by my former advisor and research group at the College of William & Mary back in the U.S. It is actually quite an interesting thing because it pulls together some information on how birds reproduce and kind of the evolutionary advantages of certain types of behaviors or lack of behaviors depending on certain environmental conditions. It pulls together some information on ecotoxicology and the effects of human disturbance in the environment, and it thinks about how the body interacts with environmental toxins and kind of mediates the effects of the habitat on reproductive success and on potentially population health and maintenance and the evolution of traits in birds. So there's a lot going on in this paper, and that makes it quite interesting to me and hopefully to you as well. So just to start off with a bit of introduction, I have talked a little bit about mercury before, I think, on the show, but just to kind of recap, it is a naturally occurring element, and we do find it out in the wild, but we particularly find it in places where humans have released it. Either they've kind of released it locally, or they have put it into things that are then released perhaps in car fumes, for example, that have been distributed over a wider range. And this is particularly associated with things like coal combustion, forest fires, and also the process of methylation of inorganic mercury, which is a form that can't really be taken up by organisms into ecosystems. Into a form that can be taken up into ecosystems. And that's the methyl mercury form. And once it is in this format, then it can be introduced into plants, it can be introduced into little invertebrates, and those things can be eaten, and then it can bioaccumulate up the food web. And this means that basically each larger organism that eats lots of the little organisms that have mercury in them can then sequester more and more of that mercury because they are eating so many of these things that have the contaminants, and then suddenly those top predators, so for example maybe an eagle, would have lots of this stuff in them because they have just taken up all of the mercury that all the little things below them in the food web have eaten first. And this obviously is not a very good thing for them. It can be quite toxic, and it can also have sublethal effects, which means that they are effects that don't kill an animal but can still have quite dramatic negative impacts. And like I said, we do tend to see this in things like eagles, but also smaller birds as well, like swallows that will eat lots and lots of little insects, each of which might have something in them, some bit of mercury that they've pulled up from the habitat. So there are lots of fish eaters and insectivorous species that might be impacted by the mercury that is naturally found in some areas, which in some places might actually be quite high, but also particularly from the mercury that humans might have introduced into the habitat at some point. Now, as I said, there are some sublethal effects that can have dramatic impacts on populations of wild animals. So for example, previous studies have shown that exposure to mercury can alter singing behavior, and as I've discussed previously on the show, singing is really important in birds for maintaining territorial boundaries, so a male will sing to keep another male from coming in. Also, it's quite important for wooing females, so if a male doesn't have quite the right song, he might not be able to get a mate, and then he won't be able to produce young. It's quite important also for just showing that you are an individual of the right species and that you are the correct sex in order for other individuals in your population to recognize you and to respond to you accordingly. So there are lots of reasons why it can be quite important to have the appropriate song in any given circumstance. So if mercury is affecting the brain's ability to produce the appropriate song, then obviously that could have quite significant impacts. Another thing that's affected is plumage patterns. So there was a study that was recently released about how mercury spills were impacting the plumage patterns of kingfishers in the U.S. And again, this is a really important signaling thing because you can use your plumage to show that you're nice and healthy. You can use it, maybe there might be subtle variations from one place to the next, and so if you don't look quite quite right for a region, then you might get into more fights, and that can cause damage later on. If you're getting in lots of fights, you can't find a territory, you're maybe getting beaten up a bit. So repercussions of a physical nature as well, perhaps. Also, there have been some suggestions that mercury contamination can potentially reduce foraging behavior or make it more difficult to forage as easily as you might have if you were otherwise quite healthy, and also to potentially reduce reproductive success. And that is actually the purpose of this paper, is to look into these potential effects on reproductive success. So I won't go into that too much more right now, because I will get into it later on. Now, cumulatively, all these sorts of things have been proposed as a serious threat to lots of different endangered species, particularly in the US, where this has been pretty well studied. So things like the rusty blackbird, a couple of different species of rail, the salt marsh sparrow, and also a thrush species have all been recognized as potentially being quite threatened by mercury. And all of these things tend to live in areas that are kind of near water or kind of are right on the water. So for example, rails, of course, they live in a similar habitat as they do here, and so they're going to be walking around in the marshes. Salt marsh sparrow obviously is in the same habitat. Things like the thrush and the blackbird would like areas that are quite near to the water, so they might be quite damp, kind of swampy, foresty areas, or areas with lots of cattails and reeds. So all of these are places where you tend to have the release of mercury and also the uptake of mercury by invertebrates in the habitat. However, despite the fact that we do tend to think of mercury contamination in relation with these aquatic environments, we also know that it can impact terrestrial species too. So those are things that might live in bogs and estuaries and wetlands, and all of that cumulatively means that actually there's a wide variety of species that could ultimately be at risk from mercury contamination, even if they live in forest habitats that are quite distant from bodies of water. And this is particularly true because you can have insects and also birds and whatever else that they might eat that would move from one habitat to another. So even if the mercury is coming from the water, it can kind of migrate onto land, if you like, and disperse inland and also upland if it is being carried by mobile organisms. And so this is why you tend to see these effects, potentially encroaching more and more if there is lots of mercury contamination away from the water and into the land, and therefore impacting all these different animals. Now, there are many ways in which mercury can impact avian reproduction. So now I'm kind of getting into the nitty-gritty part of this study and thinking about what exactly is it that these guys want to look at in the zebra finches, and why is it that they want to do this? So previous to the current study, there have been some research done on common loons, tree swallows, Carolina wrens, several other species, in which they have documented several things that could ultimately lead to some problems with reproduction. So for example, the disruption of the endocrine system, which is responsible for regulating a lot of the chemicals in the body, altering of pairing and parenting behavior. So that's kind of the sorts of things I mentioned before, maybe because it's impacting the song, maybe because it's impacting foraging behavior, whatever the case might be. It could impact how well animals are pairing up together and then producing their young and taking care of their young. There also could be direct toxicity to embryos. So we know that as a female is creating an egg, she might accidentally give it lots of toxins that she's been building up in her own body, and that could simply be too much for this developing embryo and could cause it to die off before it ever hatches, or it could cause the young chicks to die off because they're kind of sickly from the very beginning. And all the studies, or most of the studies that have been done so far, have mostly been in the wild, where you've got lots of things going on. So you can kind of correlate a couple of factors, and you can say, yes, this is a habitat with lots of mercury, and we also see that there is maybe some reduced breeding success, but it's quite hard to understand correlation and to rule out the other factors, the other things that are happening in that same environment as potential causes of the pattern that you're seeing. And to really understand how mercury is actually affecting reproductive success, and that's of course something that we need to know so that we can improve conservation and management efforts and kind of project how mercury spills and mercury contamination might impact various animals in the future. It's necessary to do dosing studies in an aviary environment. And a lot of people might kind of object to the idea of deliberately exposing animals to a toxin, but sometimes the only way for you to really understand what's going on is to do something like that in a controlled setting. And you don't even have to do it at really high levels so that you're causing the animals to suffer, because even if you're doing it at kind of lower levels, the sublethal effects, you can still get an idea of what a baseline might be. So this is, the best case scenario, and if the worst case scenario is even worse, then, you don't have to directly document it to know that it could be quite catastrophic. So what these guys did here was nothing that was really horrendously harmful up front, but it was still enough that they could get an idea of what mercury might be doing to animals out in the wild, so that they can then take that information and apply it to hopefully help those animals that are in contaminated environments. Now, when you are dosing, it's necessary also to ensure that the doses that you're giving to the birds actually reflect something meaningful. So you're not just putting this massive amount of mercury into their bloodstream and then killing them off and saying, oh yes, mercury is really bad. Because you could practically do that with anything. If you just put it in large enough amounts, you could kill off an animal. So you want to actually think, are these levels to which these animals are really being exposed, and what's the time scale over which they might have that exposure? Is this kind of a biologically relevant thing? And so what you're thinking about in this context is maybe thinking, for a food item, how much would a typical food item contain in terms of mercury? How many of those food items are eaten per day? And how many days is an animal going to be in that habitat? So then if you kind of add all of that up, then you can get an idea of how much mercury an organism is going to be exposed to on a daily basis, on a monthly basis, over its lifetime, and so on and so forth. So then you can actually kind of get this realistic quantity that's going to give you a good idea of how things are really happening out in the wild. And you also have to think about the fact that birds are kind of interesting organisms from the perspective that they are highly mobile, which means that they can be born in a site and then stay there forever if they want. They might stay there but only during the breeding season, so that they migrate elsewhere and then come back, or they might be born there and then just take off permanently. So there are all sorts of choices that you might see that these birds could make that would impact the amount of mercury to which they are being exposed and the length of time over which that exposure is happening. And that is why, in the particular study that we're looking at today, the researchers used a couple of different exposure periods to get an idea of whether there is any difference if you have these kind of different life history tactics, whether that impacts how much mercury you're going to take in, and whether that in turn impacts your reproductive success. So the current study, as I mentioned at the very beginning when I read out the title, it's looking at zebra finches, and it is using dietary methylmercury to get an idea of how zebra finches react when they are exposed either throughout their entire lives or only as reproductively active adults to mercury contamination. And the goal of all of this was to see how the mercury was impacting their reproductive success. Welcome back to The Wild Side. This is your host, Kight, and that was Frally with Songbird. And on today's show, I am talking about a recently released scientific study that looks into the impacts of mercury on the reproductive success of captive zebra finches, with the hopes of figuring out how mercury contamination in the wild might be impacting the health of various different wild bird populations. Now, just before the break, I was talking about the goals of the study, and now I want to think about the methods that the researchers used in order to look into the effects of mercury on these birds. So first of all, zebra finches are a species that I have talked about before on the show, so I won't go into them in great detail today. But basically, they're a little finch species from Australia, and they are quite popular as pets, and they're also used quite a lot in various types of scientific research. So there's a lot that's known about their life histories, about their physiology, about their morphology. They've even had their genome completely sequenced. So we know a lot about these birds basically at every level. And so they're actually quite a nice species to study in this context, because you can infer quite a lot of things given what we know about other aspects of them. So even if you aren't studying particular things right here in this given research, you know other stuff that's been done before, and so you can kind of get an idea of what the results mean and how they might have an impact on the animals kind of over the long term in the future. Now, zebra finches are a granivorous species. They aren't insectivorous, which is kind of what I keep referring to when I talk about how a lot of these birds in the wild are getting mercury in their diet. So they're eating these little invertebrates, or they might be eating fish that are then putting the mercury into their systems. So they're not quite the very perfect thing to use as a stand-in for something in the wild, except that they're actually quite easy to have in captivity, and it's very easy to dose their food with mercury. So because they're granivorous, they're just eating the regular kind of bird seed that you might go pick up at any old outdoor store, and so you can take those big bags of that and dose it with mercury and then feed it to the birds very easily. So actually it allows you to quite closely control how much mercury you're giving to them and to ensure that they actually are eating that dose. And in this case, the animals were given one of 4 different doses: 0.3, 0.6, 1.2, or 2.4 parts per million doses. And the lower 2 doses were equivalent to values that have been observed in the wild. So actually, there's a nearby contaminated site that is close to the aviary in which this work was done, and there, the researchers have previously looked at levels of mercury found in the prey items of insectivorous songbirds like the swallow and the wren that I mentioned earlier on. And so we know that these are actually things that are seen in the wild, and so they are directly relevant to the sorts of species that the researchers were hoping to apply these results to. And the higher doses were more equivalent to kind of the worst case scenario diets at contaminated sites. So maybe the bigger birds that are going to be eating the species in which some amount of mercury has already started to bioaccumulate. Now, there are 2 different parts of this experiment, so I will start with the adult exposure treatment. Now, these are birds that were exposed to mercury for 10 weeks as adults only, and after this point, blood mercury levels plateaued, and so they didn't need to keep feeding these animals more and more mercury. The animals were then randomly paired and allowed to breed for an entire year. And the eggs and chicks that were produced from these breeding events were monitored daily so that they had information about the fate of every single aspect of the entire breeding attempt. And I'll go into that in a bit more detail in a minute. The first clutch that was produced by each pair was removed so that the researchers could analyze the concentrations of mercury within the eggs, and then the birds were allowed to keep breeding, and the chicks that were produced from the following breeding efforts were then used in the next part of the study. And blood samples were collected from all of the birds each month so that the mercury levels could be monitored over time. Now, the lifetime exposure treatment, they took chicks from that previous part of the study and they paired them up, and they were allowed to mature and then given their own little space, their own little nest box, and their own little cage in order to then go on and breed themselves. So basically you've got one generation of birds that start off breeding, and these are the ones, the adults that have been exposed as mature breeders. And then you've got a second generation that is allowed to breed as well, and these guys have been exposed all of their lives to mercury because their parents were exposed and so passed on whatever mercury they might have had into the egg, and then they were also being fed by the mercury exposed individuals. So reproduction was then monitored throughout the breeding attempts as per the previous portion of the study, and across all of the different breeding attempts for both the first portion and the second portion, there were a number of different measures that were taken in order to look at reproductive performance. The first was the total number of independent offspring that were produced in a year, and an independent offspring was one that reached an age of 50 days old. The next thing was the median number of eggs in all of the clutches produced in a year. The next was proportion of eggs laid that actually made it to hatching. So there's always going to be a couple eggs in most reproductive attempts that don't turn out very well across the population, so the eggs just aren't viable to begin with, and so they never do hatch. But in this case, some of those non-hatching events might be because of the mercury, and so that's why these eggs were counted. The next thing was the proportion of chicks that hatched that then survived to fledge. So you're looking at kind of survival through the nestling period, which may have something to do with the chick's health when it hatched to begin with, but it may also have something to do with the adult's ability to feed the chicks and properly take care of them while they're there in the nest. Finally, the last thing was the number of days between fledging of one clutch and production of the next. So how long do the adults wait before they've successfully gotten rid of their first set of young before they then kind of muster their resources and have a second attempt at breeding. And usually animals that are quite fit can dive into the next breeding attempt much more quickly than animals that are maybe a bit in poorer quality or having poorer condition, they're a bit ill, whatever the problem might be, there's usually greater latency to having their next brood. And the other thing that the researchers looked at was adult mortality rates, so to see whether there were any die-offs of the parents of the young that maybe indicate that these guys are also suffering from that mercury exposure. So what did they find? Well, on average, the blood mercury levels in birds was about 13 times the dietary dose. So they were clearly accumulating all of the mercury that they were eating and over time building up more and more of it in their bodies. So for all doses, the lifetime exposed birds had slightly higher blood mercury concentrations than those that only were exposed as adults, and that's probably what you would expect given that these guys that have been exposed over their lifetimes have, of course, had more mercury around them, and so they are more likely to bioaccumulate more of it. Now, mercury also significantly decreased reproductive success, and this is potentially the more exciting result here, and that is true regardless of how you measured it. So there were several different metrics here that showed significant declines in the presence of more mercury. So for example, just to kind of give you the averages across everything, we know that the 0.3 part per million treatment had about 16% less reproductive success. The 0.6 ppm treatment had a 31% reduction. The 1.2 ppm treatment had a 42% reduction. And then the 2.4 ppm treatment had a whopping 50% reduction. So these birds at the higher end are having half as many chicks as the birds that are not being exposed to mercury at all. So that obviously would have a huge impact on animals that are living out in the wild and trying to keep their population alive. That's going to be many fewer young that survive. And then of course once the young leave the nest, there might be increased mortality at that point as well. So this actually is quite an alarming pattern if you're thinking about animals that are living in quite contaminated areas. So like I said, many different reproductive parameters were impacted. So mercury significantly reduced fledging success regardless of the amount of the dose. So this is the number of birds that you're ultimately able to get to that 50-day age and they're able to leave the nest and live on their own. They also found that birds with treatments of 0.6 parts per million and above took significantly longer to re-nest, but interestingly, birds at that very low level didn't show any difference from the control animals that weren't exposed to mercury at all. Mercury also impacted hatching success, so the lowest treatment had only slightly higher hatching success than the control, which is kind of a weird thing because that's not exactly what you would expect. You would think that as long as there's some amount of mercury, it would reduce your hatching success, and then that would kind of increase with the more mercury that you had. But in fact, the lowest treatment had slightly better success, but the highest treatment had much lower hatching success, and exactly the sort of pattern that you would expect. Neither clutch size nor adult survival were impacted, however. Now the next part of the comparison of all the various treatments and doses was to think about the two different types of exposure. So what was the difference between the animals that were impacted by mercury only as adults and those that were exposed to it throughout their lifetimes? Well, reproductive success varies significantly depending on whether or not the animals had it all throughout their lives or just as adults. In the two lower exposure treatments, animals with lifetime exposures had much lower reproductive success, but in the two higher exposure treatments, lifetime exposed birds actually had higher reproductive success. So again, that's another kind of weird pattern that the researchers certainly weren't expecting to find. They also noted that the type and level of exposure interacted to impact reproduction. So, for example, the lifetime exposed birds had a greater latency to re-nest at the two lower doses, but they had less latency at the two higher doses. And this kind of complex interaction suggests that there are potentially some really interesting things going on with the birds. Maybe it's physiological, maybe it's behavioral, maybe it's a little bit of both, but there does appear to be something going on here where the birds are potentially able to cope with the mercury in maybe an unexpected way. And I'm going to discuss that a little bit more after the break. Welcome back to The Wild Side. This is your host, Kight, and that was Ingrid Michaelson with A Bird Song. And in case you're just joining us, I will remind you that we are talking about a recent scientific study in which researchers looked at the effects of methylmercury dosing on captive zebra finches, and this was with the intention of figuring out how mercury contamination in the wild might be affecting birds that are living out in nature, with the hopes that that information could be used in conservation and management efforts. So before the break I was talking about both the methods used in the study and also the results of the research, and now I want to think about what all of those results mean and how they can potentially be applied. So the blood mercury levels that were measured in the study were similar to those that were found in the wild in previous research, so in species like Carolina wrens and tree swallows and loons and a whole bunch of different animals that have been investigated in the past. And that suggests that the treatments that were used here were accurately reflecting the sorts of conditions to which wild birds are exposed. And that's a good thing because it means that you probably can, to some extent at least, extrapolate the results from this research to what's happening out in the wild and actually use it to make some management decisions. They also found that mercury levels were impacting reproductive success in a variety of ways, and this depended on both the amount of the dose and also the length of the dose that was given to the animals. In case you didn't hear before the break, I was talking about how birds were exposed either over their whole lifetimes or just as adults, and there were 4 different doses that were given to them, kind of indicating both low levels that maybe are just slightly higher than natural levels of mercury, so just a little bit of contamination, all the way up to kind of your worst case scenario, worst possible contamination event, potentially at a point source, so maybe you've got a factory that releases a bunch of mercury into the river, for example. So the negative effects, amazingly enough, were seen even at these very lowest concentrations. So that suggests that it doesn't really take much for wild animals to begin suffering, and that suffering doesn't just have to be acute and obvious. It's not just animals dropping over dead. There can be these kind of more subtle, sublethal impacts where it's affecting their ability to reproduce, and where over time that could actually impact then the success of the population or the maintenance of certain genes and certain families of birds, if you like, over time. And so this can begin to have kind of chronic effects on the population that would then make it more susceptible to disease or to maybe an environmental catastrophe that wipes animals out all at once, or to lots of other things that maybe you would think are far removed from mercury contamination, and yet you can kind of trace the problem back to that original event that kind of weakened the overall strength of the population. So that's why it's actually quite important to think about how all these little things together can cumulatively have an impact on the birds. Now the concentrations that were used here were only about 5% of the lethal concentrations for zebra finches, and yet this actually caused 15% loss in reproductive success. So that just goes to show that if you up the amount of mercury contamination, you might expect even more catastrophic effects on reproductive success. The 2 reproductive parameters that were most influenced, which were fledging success and latency to renest, are those that are perhaps most impacted by adult behavior. And again, this suggests that it's the adults that are kind of mediating this effect of the environmental contaminants on the success of their young and on the passing on of their genes. And the reason that this might be happening is that mercury acts as a neurotoxin. And in this case, it might be reducing the efficacy of the adult breeding behaviors. The fact that adult exposed birds didn't show any reproductive suppression until about 1.2 parts per million suggests that mercury has greater impacts when the exposure is occurring during development, or that chronic exposure is more problematic than spot exposure. So unfortunately, the current study, you can't really tease apart these 2 different patterns using the current data, but the lab is looking into this possibility in more detail. And this is quite interesting because it allows them to explore whether there is this kind of lifetime accumulation of problems that then will have an effect, a noticeable effect, only once you've had enough mercury in your system over time that your body finally starts to kind of cave in, I guess, in a sense, or whether you've had this one exposure during a really critical time early on in your life that then can come back to haunt you later on when you're a breeding adult. And it is really important to investigate the distinction between those 2 different things because there are very different circumstances under which these birds might be affected out in the wild. If you've got a migratory bird, for example, that is not going to be exposed as a juvenile but then comes into a habitat later on, it may not be as impacted by the very same levels of mercury in the environment simply because it hasn't been there for its whole life, whereas something that is going to be born there and then maybe stick around could have those chronic effects coming back and affecting it after a while. And so you have to know, are these things going to have different impacts on different types of birds, birds with different types of habits, and so on and so forth. And so this kind of delving in a bit further allows you to start to figure out exactly how you could go about pinpointing the most susceptible animals and then managing to make sure that they are taken care of as best as possible. And I should also mention that previous studies have shown that mercury has significant impacts on development. So at the moment, this does seem kind of like a likely candidate, but that doesn't rule out the potential effects of kind of a chronic mercury exposure. And maybe both of these things are happening together. So there might be some interplay between kind of chronic issues and also that early developmental problem as well. Now, as I mentioned earlier, there were some kind of unexpected results in this study. One of these is the fact that lifetime exposed birds had higher reproductive success than adult exposed birds at the very highest doses. And this suggests that actually the birds might have adapted really rapidly to mercury exposure, and so this allowed them to overcome the contaminants and experience successful reproduction despite the fact that they had all of that toxin in their system. So just to kind of go through this in a bit more detail, the unsuccessful pairs in the first experiment obviously couldn't supply young to be used in the second because there were no young at all. So this was the first little bit of selection pressure. So only those young that could be produced at all, so only those young that came out of successful eggs from successful reproductive events, could go on to then have their own young in the second part of the experiment. So that weeds out one set of genes from one set of impacted birds right there. A second stage at which there would have been selection pressure is the nestling stage, during which you might have had some mortality of those less robust animals. So like I said, they were looking to see whether there were eggs that didn't hatch, and also whether there were some young that didn't make it even after they had hatched. And so if you do have young that do manage to survive the embryonic phase, but then they never are quite as healthy as some of the other birds in the population, and they therefore die out, that's another stage at which another group of genes is being taken away from the equation. And that's going to leave you only with the animals possessing the genes and the traits that are most robust in the face of mercury contamination. And so these are the birds that maybe could survive and handle that toxin and go on to reproduce their own broods. And if you have mercury acting that fast, just within a generation or two, to kind of shove the genes towards one direction of the spectrum, so you're kind of getting rid of one element of your population and keeping only the healthier, more robust element in terms of exposure to mercury, then that right there is adaptation and evolution. And so perhaps birds in the wild might actually be able to rapidly evolve the ability to deal with this particular toxin. And I'll say a little bit more on this in the future because that might be great, but it also might potentially be hazardous. So another thing to just note is that previous studies have revealed that the effects of mercury exposure vary from one zebrafinch family to another, and some families have quite a strong response and others don't have much at all. So again, this suggests that there is quite a strong kind of genetic link between how you deal with the mercury that you are being exposed to and how much mercury it takes to actually cause that kind of response. So we know that blood mercury level accumulation is also impacted by heritable genes, which again, all of this is beginning to paint the picture that evolution could really be driven by mercury exposure, not just in this species but in other species as well, as long as those guys have kind of similar characteristics where you've got variation from one animal to another and you've got this ability of the mercury to influence the amount of the variation that remains in the population. So what are the potential effects then of mercury on populations of wild birds? How can you take these findings on zebrafinches and apply that at a broader level? Well, we already know that some species are more sensitive than others. So for example, in previous studies, researchers have found that tree swallows have a much higher level of mercury bioaccumulation than Carolina wrens, despite the fact that these guys are living in the same habitat and eating some of the same food items. We also know that conditions in the aviary may have been less intense than those in the wild, so there aren't as many stressors, things like limited food, predation risk, etc., that could exacerbate the effects of mercury on avian reproduction. So that actually suggests, when you're extrapolating here, that wild birds could actually suffer more than the birds that were studied here. So the current study may kind of give you a baseline, kind of a safety zone, and then anything else in the wild is actually going to be even worse than that, and it'd be a potentially harsher impact on the birds themselves. And we do know that the reproductive impacts measured here would still have had the potential to lead to population declines, especially in small populations or in isolated populations. So again, if this is the best case scenario, it's still a pretty bad outcome, which suggests that birds in the wild could really be in trouble if the mercury is at higher levels, or if the conditions are even harsher for the animals. And this means potentially that managers could kind of use the percentage of lethal dose level that was used here as a metric for identifying habitats and populations and species that are particularly likely to be at risk. So I said earlier that they only were using about 5% of the lethal dose, and they saw about 15% reproductive decline in the birds. So you could kind of guess that anything that's 6% or more of the lethal dose would be potentially a very bad thing for animals to experience. And so once you've kind of hit that threshold, you then could trigger a bunch of management decisions and methodologies that would potentially be put in place to help animals and to help habitats. So if nothing else, this would be quite a useful starting point for identifying these things that are at risk. Now, the study also showed that lifetime exposed birds are more sensitive than those with only adult exposure, and this is roughly equivalent to the difference between kind of a sedentary or philopatric species, so animals that are spending their lives in one place or are leaving briefly but then returning to that same territory to breed, and also those that have greater dispersal distances and are more likely to migrate. So you've got these two kind of separate groups of birds, birds that are in that contaminated habitat for quite a long time, and birds that take off and are only there for a brief period. And this could be really useful for estimating which species are more likely to be susceptible. On top of this, we already know that many species of conservation concern have limited habitat. So whether that's because they prefer a type of habitat that's just not very common to begin with, or maybe it's been fragmented, maybe it's been disturbed by humans, maybe it's been contaminated by various things, mercury or otherwise. So for whatever reason, we do know that lots of species that need help are in quite small patches. And so already these are animals that we would suspect would be quite particularly at risk. And that is a nice starting point, because you know also that these are going to be the animals that are going to be stuck only in those little habitat patches. So they can't disperse too far because there is nowhere else to go to. So they're all going to use that one little bit of space, and if that one little bit of space has mercury, then you know that they're really going to be in trouble. So those are the sorts of species where you can think, oh yes, you know, these guys should be at the top of our list of species that we need to try to protect from mercury, and habitats that we need to try to get the mercury out of. Now the good news is that there was evidence of adaptation to mercury, and the same might be found also in wild populations, again assuming that they have the relevant genetic variation and that there is actually some interaction between the amount of mercury, or even the presence of mercury, and some useful thing that this genetic variation allows them to do. Now the tolerance of environmental pollutants has been documented in lots of invertebrates and in fish, and so there's no reason to think that you might not also find it in birds, so that they might actually truly possess the ability to be kind of adaptable and flexible in the face of toxins. But like I said, there might also be a bit of a drawback. So it's possible that mercury resistance might manifest as traits that are better on contaminated sites than on non-contaminated sites. And that means that migrating individuals might actually do worse in cleaner areas. So let's think about that for a minute. Because I said earlier that some of these effects, these kind of adaptive responses to mercury, might be behavioral, some of them might be morphological or physiological, or might be all of the above. So if you've got an animal that is dealing well with mercury, maybe that's because it has some different chemical that's being released in its body, or it has a different way of processing certain chemicals. Maybe it is growing slightly different feathers, or it has some other kind of morphological adaptation that's allowing it to deal with the presence of mercury. Maybe the parents are acting quite different when they are taking care of their young. Maybe you've got males that are learning different songs, but also females that are preferring different songs, so it doesn't matter if that neurotoxin activity is making the birds communicate differently. Whatever the case might be, these are all things that are going to work really well in the environment where the mercury is. But if the animals leave, then they might encounter environments in which those particular traits are actually maladaptive. So you might find that suddenly that plumage pattern or that funny song or that difference in adult parental behavior towards the chicks, all of that might suddenly not be quite so useful in an area that has no mercury at all and doesn't need you to make those behavioral or morphological or physiological adjustments. And so it's potentially problematic to think that these animals could be dispersing and suddenly encountering habitats where actually they're not doing so well at all. So it's actually just as bad for them to adapt and then move on and then suffer reproductive success as it is for them to suffer reproductive success in the contaminated habitat itself. So that makes actually things quite difficult to study out in the wild, because if this is happening, depending on the species, it very well could be, then it's going to be really hard for researchers out in the wild to really properly and comprehensively study the effects of mercury, to tease apart all these little things that are going on. Now obviously that doesn't mean that they shouldn't be trying, but it just does make it a bit trickier, and it makes the story a lot more complex. And it really goes to show you how even just a little bit of contamination from something that we're doing, you know, these burning of coal, releasing water from industrial areas, whatever the case might be, just a little bit can have these huge repercussions for wildlife. And that requires us to then do a whole lot after the fact to try to clean it up and fix it. And it actually would be much easier if we were to put regulations in place at the beginning to try to prevent that from happening at all. So that is pretty much everything I wanted to say about this particular study. I hope that you get kind of an idea of what's happening in ecotoxicology research, because actually the methods used in this study are similar to those that are employed in lots of different types of research projects on other toxins as well. And these particular birds, even though they are kind of a domestic species in a way, since we have them as pets and as research animals, they are actually a really nice indicator of what's happening out in the wild. So hopefully this information can be used by conservationists and managers also to figure out what to do about the problem of mercury and other toxins perhaps as well, to try to clean up our habitats in the wild and make them a bit safer for the animals out there. And on that hopeful note, I will leave you in the hands of Lindsay Sterling playing Song of the Caged Bird. And if there's a bit of time at the end after that, I'll be playing Peter, Sue, and Mark with Birds of Paradise.